Endotherm means that you maintain your body temperature metabolically. Endo means you're generating the heat internally. Okay? A poikilotherm is a cold-blooded animal, like in terms of vertebrates, reptiles, fish, although there are a few warm-blooded fish, reptiles and fish. Okay? Which means their body temperature fluctuates with the environment. Okay? And they're mostly ectotherms, which means they generate body temperature from the environment. They have to bask in the sun or seek shade. Okay? And their body temperature is usually at a lower set point. Any advantage of having a high body temperature? I mean, it's metabolically expensive. If I'm keeping a high body temperature, endothermically, I'm burning a lot of calories to maintain my body temperature. Right? If I'm an ectotherm, if I'm a lizard, I can kind of let my temperature drop. I'm just going to absorb heat from the environment. And then since I'm not generating metabolic heat, my caloric burn rate is lower. Like, have you ever had a pet lizard? We used to have a pet lizard in my house. My son had a pet lizard. We have had pet cats. Which one do you have to feed every day or they go nuts? The cats. You don't feed your cat for a few days, they start looking at you like really weird. They start looking like devils. They're like, oh my God, this thing's about ready to take my leg off. You don't feed the lizard for a week in a cool room, they're cool, right? Okay? So there's a definite disadvantage to being a homeotherm. A homeotherm, endotherm, you have this tremendous metabolic burn, high metabolic burn rate that you have to fulfill with fuel, food. But the advantage of a high constant body temperature is it makes your nervous system work really fast. You can imagine the advantage of that, right? No wonder mammals are the smartest animals. They have the fastest working nervous systems. Okay, that's the advantage. That advantage is enough to overbalance or overcompensate for the disadvantage of having a large appetite, right? Plus, it lets you live all everywhere in the world you want. Mammals and birds can be anywhere. Reptiles, you go to the Arctic, you don't find a whole lot of snakes up there. Okay? It's just too cold. Okay? Now check out the Siamese cat here. It's interesting, you see animals that have white fur and the tips are dark. That's a clue that the enzyme that produces dark pigmentation, which in vertebrates or mammals is always the enzyme, the pigment is called melanin. The pigment melanin that makes this fur dark is the same that makes some of your hair dark, your skin dark, your eyes dark, same pigment. And it's a multi-step pathway to make melanin. It's not just a single enzyme. But some of the enzymes are heat sensitive, which means at high heat, at normal body temperature, they don't work. They're turned off. And so, of course, where is an animal coolest? At the tips. At the tips of your, here, and the fingertips. And that's where the enzyme can work. So when you see a coloration like this, you know you've got an enzyme controlling pigmentation that's heat sensitive. Make sense? And there are some mammals, like certain Arctic foxes, that can change their fur coloration with the seasons using heat-sensitive enzymes to let their fur darken. Okay, and in this case, the enzymes are heat-stable, and they darken in the warm time of the year, so they produce dark fur that helps them to camouflage with the snowless background. And those enzymes then are inhibited by cool temperatures, and they go bleach white to get better camouflage. They change seasonally. Okay. The last slide for chapter 6, we're moving to chapter 7, has to do with a discovery by Joe Keck and Bilian Zhang in the early 1980s at Colorado University at Boulder, the discovery of enzymes that are not proteins. These are known as ribozymes. A ribozyme is an RNA enzyme. I talked about these from day one in this course a little bit. And probably, I think there's a good chance, many people hypothesize that in the history of life, ribozymes precede, in the history of life, protein enzymes. So they are the early version of enzymes. They're like enzyme 1. Okay? Enzyme version 1. There's a guy at, I think he's retired now, Henry Knoller. He was at that hippie university, UCSC. They used to have, in the old days at UCSC, UC Santa Cruz, no grades. How about that, man? You pass or no pass. Of course, it was hell to get med school, because then you apply to med school, and it's like, well, what's your grade point? I don't have one. So they had to kind of figure out a way to, that's why eventually the system didn't work. But it's kind of a, it's to make a kind of a learning environment where you're stress-free, like peace and happiness, okay? The hippie university. It is one of the top universities in the world. I mean, a lot of the bioinformatics algorithms to use to mine genetic data are developed at UCSC. They got a great astronomy, so they world-class scientists. This guy here, he was the one the first one to realize that the ribosome, you know what the ribosome does, right? Remember the ribosome? What does the ribosome do? What's the process that it drives? Starts with a T. Translation. Translation is, remember, is mRNA to protein. Okay. And when you link amino acids to form proteins, what's that bond called that links amino acids together? That the ribosome catalyzes? What kind of bond holds amino acids together in a protein? It's called a peptide bond. Okay? Remember, it's a dehydration synthesis reaction. Just trying to bring up some words you have to think about pretty soon, right? Okay. So the ribosome is a large, complex cellular machine that has enzymatic capacity. It can catalyze the formation of these bonds between amino acids. And remember, what two types of molecules make up ribosomes? They're made out of proteins and ribosomal RNA. So the two structures that make up ribosomes are proteins, and I think there's like 80 proteins or something like that. I forget the number now. And ribosomal RNA. Okay, it's another type of RNA that's something beyond. Most introductory students think about RNA only as messenger RNA. There's a lot of other RNA versions that do other things. Okay, this is a part of the ribosome structure. When scientists first thought about how ribosomes catalyze peptide bond linkage in the synthesis of proteins, they just assumed that the catalytic part of the ribosome was the protein, because biological catalysts are proteins. But when Noller removed all the proteins from ribosomes, the damn thing still built peptide bonds. So it's the ribosomal RNA of the ribosome that is a ribozyme. Okay? Now his work was criticized because, and rightfully so, the critics said, well, it's when you remove the proteins from this machine, are you sure you got them all out? Because you have to, you have every instrumentation has detection limits, whether it's wet chemistry or some spectrophotometric method, and he couldn't say for certain that all the proteins were removed. So the critics argued maybe trace proteins left in the ribosomes were present enough to form these peptide bonds. That's a valid criticism. So what Keck and Zhang did is they went in the laboratory and they synthesized a whole bunch of RNA molecules at random. They just linked the whole, stuff a whole lot of them together. Okay. And then they wanted to see if any of them had catalytic activity. Could they form a peptide bond? So they attached to the 5' end of the RNA molecule a disulfide bridge hooked to a phenylalanine. Okay, and then they added into reaction mixture some methionine linked to AMP, so it kind of has the energy to form a peptide linkage, attached to a substance called biotin, which acts like a tag. With biotin, you can grab things in a chromatography column. It's a common method to isolate structures. They had all these numerous reactions set up, I don't know how many, where each reaction had a unique RNA molecule. The RNA sequence was unique. They wanted to see, could a particular, of all these thousands of RNA molecules we synthesized, could one of that batch catalyze the linkage of methionine to phenylalanine, form a peptide linkage? So you let the reaction occur, and then you take these and you pour them through a chromatography column. And in the column is a substance called streptavidin, which will lock onto biotin and grab it. Okay? And if the RNA could form the linkage, if the RNA could catalyze this peptide bond formation, just like a ribozyme, the streptavidin in the column would capture the whole RNA molecule. But all those that were non-catalytic, they just washed right through the column. Does that make sense? Am I going too fast? And they found some that, some were captured by the beads in the column, because the captured RNA, because the linkage between phenylalanine and methionine catalyzed the RNA carried the tag, and the tag trapped it in the column. Okay, and so they knew that some RNAs could, pure RNA, there was no protein contamination, could truly function as a ribosome. That was kind of a landmark. I mean, these guys got a Nobel Prize for that. Okay, and that's an important discovery, because since this work in the early 80s, the role of RNA in cells is the recognition of the role of RNA has become exponentially in terms of the amount of work that RNA performs in cells. There's a lot more than we early textbook ever dreamed of. If you look at early textbook, they talk about ribosomal RNA, transfer RNAs, messenger RNA. They might talk about a little bit about the spliceosome and a nucleus that helps to remove introns from premium RNA, but now there's all types of RNA machines. This might be relics of the early RNA world, because remember I talked about early in this class, when you look at this central dogma, DNA to RNA to protein, that all life uses to convert the genetic language into the machines of the cell. Okay, this of course is called transcription, converting the DNA gene into an RNA transcript, and then translating the RNA language into a protein language. These processes depend upon protein enzymes. What is the main? Now there's a whole cast of enzymes. What's the main enzyme that converts DNA to RNA? It's called RNA polymerase. Let's just talk about this one, for example. There are many proteins involved here.